You mail the tube on a Tuesday. It takes about 30 seconds, spit, seal, drop it in the mailbox, and then you forget about it for 6 weeks. When the results arrive, most of what you see is predictable. Irish, German, a little Scandinavian. The pie chart matches the family stories. And then, in a small sliver at the bottom, a label you did not expect. Indigenous American. 2%. Maybe 3%. No name, no tribe, no explanation. Just a number. And you have no idea where it came from. Neither does your mother. Neither does anyone else in the family. And it turns out, you are not alone. Millions of Americans have done exactly this. They have sealed a small plastic tube, sent it through the mail to a genomics company, and waited for a chart that was supposed to tell them who they are. The ritual has become so common, it barely registers as unusual anymore. Somewhere in the neighborhood of 40 million people have submitted consumer DNA tests in the United States alone. It is the largest voluntary genetic database in human history, assembled one plastic vial at a time. Most of the results, for most people, confirm what was already known. The Irish find Irish. The Italians find Italian. The Eastern Europeans find the borders their grandparents described, redrawn in percentages instead of maps. The tests are, in the main, satisfying in a quiet way. They give people the sense that they are looking at themselves from the outside. A biological receipt for the family they already had. But then there are the results that do not fit. Across the country, people with no known Native American ancestry are receiving small percentages labeled Indigenous American. A woman in Ohio whose grandparents all came from Poland and Romania. A man in Michigan who can trace every branch of his family tree to Northern Europe without interruption for 5 generations. A family in Virginia whose oral history contains nothing, not a rumor, not a whispered story, not a single suggestion of any native ancestor. And yet the test comes back and says, something is there. The easy explanation, the one that most people reach for first, is that the test is wrong. An algorithm error, a quirk in the database, a case of the company confusing one kind of DNA for another. And for some people, that explanation may be exactly right, but it does not explain everything. Because when researchers looked at the data, not at individual results, but at patterns across tens of thousands of people, what they found was not random noise. It was geography. A 2015 study published in the American Journal of Human Genetics, using data from 23andMe, analyzed nearly 149,000 self-identified European Americans across the United States. The results were not uniform. In Louisiana, up to 8% of European Americans showed at least 1% Indigenous American genetic ancestry. The figure was lower in the upper Midwest, lower still in the Northeast, and almost absent in regions where colonial contact with native populations was minimal or heavily documented. The pattern did not look like a bug in the software. It looked like history. The same study found something else. Of more than 5,000 self-identified African Americans in the dataset, over 22% showed at least 1% Indigenous American ancestry. That figure had been largely invisible in the historical record. It had not disappeared from the population. It had simply been undiscussed. This is the problem at the center of a very modern puzzle. Millions of Americans are receiving a result they cannot explain. Some of them have family stories, a grandmother who was Cherokee, a great-grandfather who was Comanche, a branch of the family that was always described as part Indian, and the test confirms that something is there. But others have no story at all. Nothing in the oral record, nothing in the census documents, nothing in the church registers, and yet the DNA keeps returning the same small signal. And here is what makes it stranger. Other people with detailed, documented, multi-generational family stories of Cherokee or Ojibwe or Lakota ancestry, take the same kind of test and receive nothing. 0. The pie chart comes back entirely European or entirely African American, with no indigenous marker at all. They know the story is real. They have documentation. They have names and dates and photographs. But the algorithm does not see it. The contradiction runs in both directions. Some people get a result they cannot explain. Others cannot get a result they believe they deserve. And both of these facts, it turns out, are clues to the same deeper story. The first instinct is to blame the technology. And there is something to that. Consumer ancestry tests work by analyzing a subset of your DNA, somewhere between 400,000 and 700,000 locations in the genome. Out of a total of roughly 3 billion base pairs, they are not reading your entire genetic code. They are reading a carefully selected sample of it and then comparing that sample against a reference panel, a database of people whose ancestry is considered known, grouped by geographic origin, against which your DNA is measured. The category labeled Indigenous American on most consumer tests is built from that reference panel. And here is the first technical problem. The reference panels used by the major companies are not built primarily from enrolled members of North American tribes. They are built mostly from people in Latin America and the Caribbean who self-identify as having indigenous ancestry. The genetic diversity of the Ojibwe, the Lakota, the Haudenosaunee, the Cherokee, the actual nations of North America, is dramatically underrepresented. Many tribes have declined to participate in commercial genetic research, and for reasons that go back much further than a single policy decision. So the algorithm is, in part, comparing your DNA against a proxy. It is asking whether your genetic markers resemble the markers of indigenous Latin American populations, and using that resemblance as a stand-in for North American tribal ancestry. This means that a person with genuine Lakota ancestry might get a weak or absent result, because the Lakota genome is not well represented in the reference panel. And a person with no North American tribal ancestry at all might get a small signal, because something in their DNA resembles the Latin American indigenous reference population in a way the algorithm interprets as ancestry. But that still does not explain everything, because there is another complication, one that goes deeper than reference panels and algorithm design. It has to do with how DNA is actually inherited. When you receive genetic material from your parents, you do not receive a clean 50% from each. The process of recombination, the shuffling of chromosomes during the formation of reproductive cells, is essentially random at the level of individual segments. Over generations, this randomness compounds. By the time you reach a great-great-grandparent, you are not guaranteed to have inherited any detectable DNA from that individual at all. Statistically, by the 5th generation, there is roughly a 50% chance that you carry zero detectable genetic material from any single ancestor. The DNA may simply not have been passed down to you. What this means in practice is that two siblings who share the same great-great-grandmother, the same Cherokee great-great-grandmother, the same real documented ancestor, can take the same consumer test and get completely different results. One might show 2% Indigenous American ancestry, the other might show 0. Neither result is wrong. They are both accurate reflections of what was randomly inherited and what was not. This creates a situation where the absence of a result does not disprove an ancestor, and the presence of a result does not always confirm one. The test is measuring a statistical shadow of ancestry, not ancestry itself. The shadow appears in some descendants and not in others. Depending on the random outcomes of chromosomal shuffling across multiple generations, and yet the signals that appear in the 23andMe data clustered in the South, stronger in Louisiana than in Minnesota, present at statistically meaningful levels in both European American and African American populations, are not random, they are patterned. And a pattern implies a cause. The simplest version of the explanation is historical. People mixed. European colonists and Native Americans had children together. African enslaved people and Native Americans had children together. These relationships happened across centuries, in places that were often distant from official record keeping, in circumstances that gave families strong incentives to conceal certain kinds of ancestry. The DNA survived even when the story did not. That is part of the answer, but only part. Because when geneticists looked more carefully at the signals, not just their presence, but their structure, their distribution, the specific markers they involved, some of them pointed to something older than any colonial encounter, something older than the European arrival in the Americas, something that traced back not to a hidden grandmother in the 1800s, but to the earliest movements of human beings across the planet. The test result showing 2% Indigenous American ancestry at a kitchen table in Ohio is not just a family mystery. It is, in some cases, a message from a migration that happened before written history existed. the people who crossed into the Americas were not genetically isolated from the rest of humanity. They came from populations that were connected to other populations, and those ancient connections left traces that look to a consumer algorithm in the 21st century, like a percentage. But what those traces actually mean and where they actually came from turns out to be one of the more genuinely complicated questions in the science of human origins. The geography of the results is not an accident. The signals cluster in places where certain kinds of history unfolded. And understanding why requires going back to the actual history, not the sanitized version, and not the family legend, but the documented, complicated, frequently uncomfortable record of what happened when the populations of an entire hemisphere were brought into contact with one another for the first time. The test says 2%. The family has no story. But the ground does. The geographic clustering in the 23andMe data tells you something important before any deeper explanation is attempted. Louisiana is not random. The South is not random. The states with the highest rates of detectable Indigenous American ancestry in European American genomes are precisely the states where the history of contact, trade, intermarriage, and deliberate erasure was most concentrated. That is not a coincidence. That is the biological residue of specific events. But the story is harder to tell than the test result suggests. Most Americans learned a version of colonial history that treated Native Americans as peripheral figures, people who were present at first contact, who receded as European settlement expanded, and who mostly disappeared from the story of America's founding. That version is not only incomplete, it is, in the places that matter most for understanding the DNA data, simply wrong. In the early centuries of European colonial presence in North America, the interior of the continent was not a European world. It was a world of indigenous nations and the Europeans who moved through it, traders, trappers, missionaries, soldiers, and settlers. Often did not live at the edge of native society. They lived inside it. They learned native languages. They married native women. They had children. Those children were documented sometimes and erased from the record at other times, depending on what was useful to the families and communities involved. The signals in the data are not scattered randomly across the American population. They are concentrated in the descendants of the people who lived in those contact zones. And the test, for all its imperfections, is picking up the biological echo of a history that the written record was designed to obscure. But there is another layer beneath the historical mixing, another set of signals that point not to a colonial trading post in the 18th century, but to migrations that happened on a completely different timescale, signals that predate any historical record, signals that connect the question of why your test says 2% Indigenous American to the larger and still unresolved question of where the first Americans came from and who they were related to on the other side of the world before they ever became American at all. That question has an answer that mainstream science largely agrees on, but it has details, persistent, genuinely strange details that have never been fully resolved. And those details are where the story gets considerably older and considerably stranger than any family secret. There is a church ledger in South Carolina, brittle at the edges, that records a marriage from the year 1754. The groom is listed by name, a Scots-Irish trader, his occupation noted, his parish recorded. The bride is listed as Indian woman, no name, no nation. No further description. The union produced children. Those children were baptized. Their names appear in later records as fully European. By the third generation, the Indian woman had vanished from the family's official story, replaced by a genealogy that ran cleanly back to the British Isles. She left no name. But she left Na. This is the kind of erasure that the 23andMe data is in some cases reversing. Not dramatically, not personally, not with the resolution anyone would want, but statistically, at the population level, the biological record is reasserting something the paper record tried to remove. And to understand why the signals appear where they do, it helps to understand the specific history that produced them. The mixing of European, African, and indigenous populations in what is now the United States did not happen uniformly, and it did not happen by accident. It followed the geography of contact, the economics of trade, and the political pressures of colonial expansion. In the Southeast, where the signals are strongest, the history is also the most layered. The French colonial presence in Louisiana and the lower Mississippi Valley was, from its earliest decades, deeply entangled with indigenous nations. The Natchez, the Caddo, the Houma, the Choctaw, the Chickasaw, these were not peripheral actors in the colonial story. They were trading partners, military allies, sources of food, guides through terrain the French could not navigate alone. French fur traders, missionaries, and soldiers lived alongside and among these nations for generations. Intermarriage was not unusual. Children of mixed French and native parentage occupied a recognized social position in early Louisiana, a position that would later be erased as American racial categories hardened. The Scots-Irish traders who moved through the Cherokee and Creek territories of the Southeast operated under similar conditions. The deerskin trade that ran through the Appalachian interior in the 17th and 18th centuries required sustained relationships with native communities. Traders who were successful were often traders who had native wives, women who served as translators, cultural mediators, and social anchors within communities where an outsider alone would have been useless. The children of those unions were often raised in the native community, educated in two worlds, and documented in neither with any particular consistency. None of this was secret at the time. The mixing was visible, acknowledged, and in many contexts, economically logical. What changed was the political climate. As the 19th century progressed and American expansion moved inland, the pressure on native populations intensified. The Cherokee removal of 1838, the event now known as the Trail of Tears, forced approximately 16,000 Cherokee people from their ancestral lands in Georgia, Alabama, Tennessee, and North Carolina westward to what is now Oklahoma. The death toll during the forced march is estimated at 4,000 people or more. But the removal did something else besides displace a population. It created a powerful incentive for families of mixed Native and European ancestry to choose a side. Families that could pass as white did. The risk of being categorized as Native American in the years surrounding the removal was not abstract. It was the difference between keeping your land and being marched at gunpoint into the winter. Census records from the 1830s and 1840s in the Southeast show a striking pattern: the number of people identifying as Native American or mixed race plummets in exactly the areas where removal was most actively enforced. People did not disappear. They reclassified. A Cherokee grandmother became Spanish. A Creek great-grandfather became French. A woman who had been listed as half Indian in an 1820 census appears in the 1840 census as white. The paper record accommodated survival. The biology did not change. This process of erasure was not unique to the Trail of Tears. It repeated in different forms across different regions and different eras. The 1887 Dawes Act, which required Native Americans to enroll on official tribal rolls in order to receive allotments of land, created another pressure point. Families who could not prove enrollment or who chose not to attempt it for fear of the consequences fell off the official record. By the early 20th century, there were people with 4 Native American grandparents who appeared in every official document as white, because the alternative had, at some point in the recent past, been too dangerous. The DNA did not get the memo. What the 23andMe geographic data is showing, in part, is the biological persistence of these erased histories. The signal is stronger in Louisiana because the French colonial period produced more mixing that was later covered over. The signal is stronger in the Southeast because the contact between Scots-Irish traders and Cherokee and Creek women was extensive and sustained, and because the removal created the most powerful incentive to erase it. The signal appears in African American populations at rates that reflect a different but parallel history, one in which enslaved people and Native Americans shared territory, sometimes shared escape routes, sometimes intermarried in the spaces outside the legal reach of the plantation system. The 5 civilized tribes, Cherokee, Choctaw, Chickasaw, Creek, and Seminole, each held enslaved black people before the Civil War, and each had significant populations of black freed people who remained in Indian territory after emancipation. The genetic signals moving between African American and Native American populations in the data reflect centuries of contact, cohabitation, and kinship that the official racial categories of American history were never designed to capture. But not everyone who receives an Indigenous American signal on a consumer test has a hidden colonial ancestor. The history of mixing explains the clustering, the geographic patterns, and a significant share of the signals that appear in populations with no family knowledge of native ancestry. It does not explain all of them, because some of the signals appear in places and populations where the documented history of colonial mixing is thin. Some appear in people whose family trees go back to colonial New England, where the history of contact was different, more violent, less economically collaborative, less likely to produce the kind of sustained intermarriage that characterized the Southeast. Some appear in people who have traced their genealogy with professional rigor and found nothing that could account for the signal within any historically plausible time frame. For those cases, the explanation is not in the colonial record. It is in something much older. There is a version of this story that begins not in 1754 with a nameless bride in a South Carolina church, but approximately 15,000 years earlier, when a population of human beings who had spent generations in the cold intersection between Asia and North America finally moved south into a hemisphere that had never seen a human footprint. The founding population of the Americas, the ancestors of every Native American nation that would ever exist, did not arrive as a blank genetic slate. They carried with them the genetic legacy of the populations they had descended from, the populations they had lived alongside, and the migrations that had shaped them across tens of thousands of years before any of their descendants ever saw the Mississippi River or the Andes or the Amazon basin. And some of the signatures in that founding genetic package turn out to overlap, in small but measurable ways, with signatures that modern genomic algorithms associate with the Americas. The signal in your test may not be pointing to a great-great-grandmother. It may be pointing to a population movement that ended before agriculture existed anywhere on Earth. And that is where the story stops being about colonial history and starts being about something that is. In the most literal sense, prehistoric. The history you were not taught in school explains part of the DNA result. But the part that cannot be explained by history requires a different kind of investigation entirely. One that takes place not in church ledgers or census records, but in ancient bones, frozen permafrost, and the machinery of a genomics laboratory processing samples from people who died thousands of years before anyone thought to write anything down. The mixing that produced the patterns in the data was real, it was extensive, and it was deliberately hidden. That is one answer. But beneath that answer is another one, older and stranger, and it begins on the other side of the Bering Strait. Picture a landscape that no longer exists. 15,000 years ago, the shallow seafloor between what is now eastern Siberia and western Alaska was dry land. Not a narrow crossing, not a precarious ice shelf, but a broad subcontinent, sometimes called Beringia, that stretched nearly 1,000 miles from north to south. It had rivers, grasses, grazing animals, and the kind of cold, open ecology that a certain kind of human population had learned to survive in. For a period of perhaps several thousand years, a group of people lived there, not passing through, living there, hunting the megafauna, following the seasons, existing in relative genetic isolation from both the Asian populations to the west and the empty Americas to the east. When the climate shifted and the glaciers retreated enough to open corridors southward, those people moved. They moved into a continent that had never known a human being. And everything that followed, every nation, every language, every civilization, every agricultural system, every city, every piece of art, every burial mound, every carved stone, descended from that founding population and the migrations that came after. This is the part of the story that mainstream science largely agrees on. The timing is still debated at the margins. Some evidence suggests human presence in the Americas as early as 30,000 years ago, well before the commonly cited dates, though this evidence remains contested. The Anzac I child, a Clovis-era infant whose remains were found in Montana and whose genome was published in 2014, dates to approximately 700 years ago, and is confirmed as ancestral to many modern Native American populations. The USR-1 infant from the Upward Sun River site in Alaska, whose genome was published in 2018, dates to roughly 11,000 and 500 years ago and helps define the genetic profile of the Beringian founding population. What the ancient DNA confirms is that the people who crossed into the Americas were genetically distinct from modern East Asian or Siberian populations, not because they came from somewhere else, but because they had been isolated long enough in Beringia to diverge. They were not a random sample of ancient Siberians. They were a specific population, shaped by thousands of years of separation, carrying a genetic signature that would eventually become the defining mark of the indigenous peoples of two continents. And here is where the story begins to complicate itself. In 2012, a genome was published that changed the way scientists understood the relationship between Native Americans and the rest of humanity. The sample came from a 24,000-year-old individual found near the village of Malta in Siberia along the Belaya River. The individual is known in the scientific literature as MA1, or the Malta boy, though the sex assignment has since been revised. What the genome revealed was unexpected. MA1 did not belong to any of the East Asian lineages that most researchers had assumed were the primary source of Native American ancestry. Instead, MA1 belonged to a lineage now called Ancient North Eurasian, a population that appears to have been widespread across northern Eurasia during the last ice age, but left no clear descendants among any modern Siberian or East Asian population. The Ancient North Eurasian lineage is, however, detectably present in two very different groups of modern people: Native Americans, who carry it at levels estimated between 14 and 38% depending on the population, and Western Eurasians, including modern Europeans, who carry it at lower levels through a different route of descent. What this means, in practical terms, is that Native Americans and Europeans share a common ancestral component that has nothing to do with post-Columbian contact. It is not a signal of mixing after 1492. It is a signal of shared ancestry from a population that existed and then largely vanished from the genetic record tens of thousands of years before any European set foot on American soil. The implications for consumer DNA testing are real and largely under-discussed. When a genomic algorithm looks at the DNA of a person of primarily European ancestry and compares it against a reference panel that includes indigenous American populations, it may detect the ancient North Eurasian signal that Europeans and Native Americans share. Depending on how the algorithm is calibrated, it may classify a portion of that shared ancient ancestry as indigenous American, not because the person has a Native American ancestor in any historical or genealogical sense, but because the deep genetic overlap between these two distant populations creates a resemblance the algorithm cannot always cleanly distinguish. This is not a flaw in the test so much as a fundamental challenge in population genetics. Human populations did not develop in neat, sealed compartments. They mixed, separated, drifted, went extinct, rebounded, and mixed again over timescales that make the entire history of European colonialism look like a recent footnote. The categories that consumer tests use, European, Indigenous American, East Asian, African, are useful approximations of real genetic clusters, but they are approximations. The underlying reality is a set of overlapping and partially connected ancestry streams that stretch back far beyond the invention of any identity category. The Kennewick Man case is instructive here. When the skeletal remains of a man who died approximately 8,500 years ago were found along the Columbia River in Washington state in 1996, the skull's morphology, its shape, proportions, and features, was described by some researchers as resembling Polynesian or Ainu populations more than modern Native Americans. This observation was used, in some quarters, to argue that the earliest Americans were not ancestral to modern native peoples, that there had been earlier, different populations that were later displaced. The ancient DNA resolved the question. A genome analysis published in Nature in 2015, led by Eske Willerslev at the University of Copenhagen, confirmed that Kennewick Man is most closely related to modern Native Americans, specifically to the Confederated Tribes of the Colville Reservation in Washington state, whose territory overlaps with where the remains were found. The skull morphology had been misleading. The DNA told a different story. Kennewick Man was not a separate population. He was an ancestor. But the resolution of the Kennewick Man debate did not close every question in early American genetics. It answered one. it quietly highlighted others. Because if the founding Native American population had its roots in Beringia, and if that population carried ancient North Eurasian ancestry that it shared with Proto-European populations through a common Siberian ancestor, then the genetic overlap between modern Europeans and modern Native Americans is real and ancient and has nothing to do with the colonial period. A small percentage of that overlap, misread by an algorithm, could appear as indigenous American ancestry in a person with entirely European family history. This is, in some cases, probably exactly what is happening. And the companies know it. The calibration of the reference panels, the threshold for calling a percentage, the way the algorithm handles ambiguous signals in the ancient Eurasian ancestry space, these are all technical decisions that have real consequences for the results people receive and the stories they build around those results. But ancient shared Eurasian ancestry explains a portion of the signal, not all of it. And the ancient North Eurasian component is not the strangest thing hiding inside the genetics of the first Americans. Because there is one specific maternal lineage, a branch of the human mitochondrial family tree that appears in Native American populations, but whose Old World relatives have never been clearly identified. A lineage that seems to arrive in the Americas fully formed, without a clear parent population in any currently living population anywhere in the world. A lineage that has, over the past 2 decades, been at the center of some of the most dramatic and ultimately cautionary debates in the science of human origins. Its name is haplogroup X2A, and its presence in the American genome is something that mainstream science has an answer for, and also something that mainstream science has never quite finished explaining. The Malta genome was a landmark. The Kennewick Resolution was a landmark. The mapping of the Beringian founding population was a landmark. Each one answered a question and opened a smaller, more specific one. And haplogroup X2A is one of those smaller, more specific questions, a single branch on the human family tree whose story leads somewhere unexpected, and whose resolution required demolishing one of the most ambitious alternative theories in modern archaeology. The genetics of the founding Americans is, in the broad strokes, well understood. In the fine details, it remains one of the most genuinely active debates in the science of human prehistory. And the fine details are exactly where the story of why your DNA test says what it says gets both more honest and more strange. Open any textbook on mitochondrial DNA and you will find the same basic diagram, a branching tree of maternal lineages, each branch representing a mutation that occurred in a specific woman at a specific point in deep time and was then passed down through her daughters, and their daughters, and their daughters' daughters, in an unbroken maternal line to the present. The tree is a record of human movement. Every time a population separated from another, migrated to a new region, and stopped interbreeding with the group it left behind, the mutations in its mitochondrial DNA began to accumulate independently, producing a new branch. The mitochondrial lineages found in Native American populations are labeled A, B, C, D, and X. The first 4, A, B, C, and D, have clear Asian relatives. Their parent lineages exist in modern Siberian and East Asian populations, and the evolutionary pathway from the Old World to the New World is traceable, if not perfectly mapped at every step. They entered the Americas through Beringia. The evidence for this is strong. And then there is X. Haplogroup X is not an American lineage. It is found across the Old World, in the Near East, in the Caucasus region, in Central Asia, in small frequencies across Europe, and among the Altai people of southern Siberia. It is an ancient lineage with a wide distribution, but within the Americas, only a specific subclade of X is present. Not X in general, X2A specifically, a branch that carries a distinct set of mutations that set it apart from every Old World X lineage that has ever been found. The numbers are striking in their specificity. Haplogroup X2A appears in up to 25% of Algonquian-speaking populations. It is present in approximately 15% of Sioux mtDNA samples. It appears in the Ojibwe, in the New-Cha Nulth, and several other nations of the Great Lakes and northeastern regions. In the overall Native American population, its frequency is lower, around 3%, but its geographic concentration in the north and northeast of the continent gives it a distinctive pattern. It is not evenly distributed across the Americas. It clusters. And it is old. The remains of Kennewick Man, the individual found along the Columbia River who died 8,500 years ago, carried haplogroup X2A as his mitochondrial lineage. Pre-Columbian skeletal remains in multiple sites have been tested and found to carry X2A, confirming that this is not a lineage that arrived with European colonists after 1492. It was here before anyone arrived from Europe. It is genuinely Native American in the historical sense. The question is where it came from before that. If you go looking for the Old World ancestor of X2A, the specific variant of X that gave rise to X2A through the mutations that define the subclade, you find something strange. You find that it is not there. The intermediate form. The version of X2 that should exist between the widespread Old World X2 and the American X2A has never been found in any living population anywhere in the world. Not in Siberia, not in the Altai, not in the Caucasus, not in the Near East, where X2 is most common. Not anywhere. This is known in the scientific literature as the problem of the missing link. Every other Native American mitochondrial haplogroup has identifiable Old World relatives, imperfect, partial, sometimes distant, but traceable. X2A does not. Its parent seems to have vanished. There are several ways to interpret this absence. The most straightforward, and the one that the majority of geneticists accept, is that the population carrying the intermediate form went extinct. Human populations went extinct all the time during the Pleistocene. The Ice Age was not kind, and large-scale population collapses that wiped out entire genetic lineages were common. The founding population that carried Proto-X2A into Beringia may have survived long enough to cross into the Americas and then disappeared in the Old World, leaving no modern descendants. The Americas preserved what Siberia lost. This explanation is not implausible. It is, in fact, probably correct. Jennifer Raff and Deborah Bolnick made this argument carefully and rigorously in a 2015 paper in the journal Paleo-America, reviewing the evidence and concluding that the most parsimonious interpretation of X2A's distribution is a Beringian origin from a founding population that subsequently went extinct in the Old World. The absence of intermediate forms reflects the catastrophic population history of Ice Age Siberia, not the existence of an undiscovered migration route. But before that explanation gained its current dominance, the missing link created an opening, and into that opening walked a theory that was, depending on your perspective, either one of the most creative hypotheses in modern American archaeology or one of the most spectacular examples of scientific overreach. The theory proposed that X2A did not come from Siberia at all. It proposed that haplogroup X2A entered the Americas from Europe, carried by Ice Age people who crossed the Atlantic along the edge of the pack ice, somewhere between 21,000 and 17,000 years ago, making landfall in the northeastern part of North America thousands of years before anyone crossed Beringia. The theory came from two serious researchers, Dennis Stanford, a Paleo-Indian archaeologist at the Smithsonian Institution, and Bruce Bradley, an archaeologist at the University of Exeter. They did not publish their ideas in a fringe magazine. They published a book, Across Atlantic Ice, in 2012 through the University of California Press. They presented their hypothesis at academic conferences. They had careers built on legitimate, peer-reviewed work. And they had, in addition to the genetic argument, an archaeological argument that was genuinely interesting. The stone tool technology of the Clovis culture, the earliest widely recognized archaeological culture in North America, dating to approximately 13,000 years ago, shares certain features with the Solutrean culture of Ice Age Europe. The Solutreans lived in what is now France and Spain between roughly 22,000 and 17,000 years ago. Both cultures used a technique called overshot flaking, a particular method of shaping flint tools by removing thin, flat chips from one face of the stone across its entire width that is not commonly found in the archaeological record between these two cultures geographically. Stanford and Bradley argued that the similarity was not coincidence. They argued it was contact. If you stand in front of a museum case displaying a Clovis point and a Solutrean point side by side, the resemblance is striking enough to stop you. The shapes are different in detail, but similar in execution. the same elegant thinning, the same careful control of the flaking pattern, the same functional geometry. Whether that resemblance reflects a shared origin or independent invention is a question that requires more than a museum case to answer. But it is a real resemblance, and it was real enough to anchor a serious academic debate for several decades. The Solutrean hypothesis was not accepted by the mainstream. Criticisms were extensive and substantive. The chronological gap between the end of the Solutrean culture and the beginning of the Clovis culture is significant, roughly 2,000 years, in which no Solutrean-derived archaeological signature appears anywhere in the archaeological record of the Atlantic. No Solutrean maritime technology capable of sustained Atlantic crossing has ever been found. The geographic distribution of Clovis sites does not match the predicted pattern of an Atlantic entry. The oldest Clovis sites are not in the northeast, where an Atlantic crossing would have arrived, but are distributed widely across the continent. And most geneticists were skeptical of the haplogroup X2A argument from the beginning, because X2A is not found in any ancient or modern European population. But the hypothesis lived on, in part because the absence of the intermediate X2A form in the Old World kept the question technically open. As long as the missing link was missing, the Beringian origin of X2A was an inference, a strong, well-supported inference, but an inference nonetheless. And then the ancient DNA arrived. Not from the Americas, from Europe. In 2023, researchers published genomic analyses of actual Solutrean-era individuals, people who had lived in Ice Age Europe during the period when the Solutrean culture was active. Their genomes were sequenced, their ancestry was mapped, and the result was unambiguous. Solutrean individuals showed no relationship whatsoever to Native American populations. The genetic connection that the hypothesis required did not exist. Not a trace of it. The people who made the tools in France and Spain 20,000 years ago were not the ancestors of the people who made the tools in New Mexico 13,000 years ago. Michael Waters of Texas A&M University Center for the Study of the First Americans stated the conclusion directly. Recent genetic work on Solutrean human remains shows they are not related in any way to the indigenous peoples of the Americas. The hypothesis was not fringe pseudoscience. It was proposed by credentialed researchers at major institutions, and it was taken seriously enough to generate decades of peer-reviewed debate. That seriousness is what makes its final resolution instructive. The lesson is not that bold hypotheses are wrong. The lesson is that genetic signals, including haplogroup signals, can be misread when the right ancient DNA has not yet been tested. The absence of the intermediate X2A form looked, for a while, like a door. The Solutrean ancient DNA closed it. X2A almost certainly arrived in the Americas through Beringia, in a founding population whose Old World relatives later went extinct. The missing link is missing, because extinction is permanent. The theory that tried to explain it differently failed when confronted with direct evidence from the proposed source population. But haplogroup X2A is not the only unexplained genetic signal in the story of the first Americans. Because while the Solutrean debate was unfolding, another discovery was being made in a cave in Brazil, a discovery that pointed not to Ice Age Europe, but to somewhere even more unexpected, and that raised a question about the peopling of the Americas that has not yet been answered. The Lagoa Santa region of Minas Gerais, state in Brazil, is not a dramatic landscape in the way that the American Southwest or the Peruvian highlands are dramatic. It is limestone karst country, low hills, shallow caves, dry scrub, a terrain that looks, from a distance, almost unremarkable. But the caves of Lagoa Santa have been producing human skeletal remains since the 19th century. When the Danish naturalist Peter Lund excavated dozens of ancient individuals from the cave floors and shipped their bones to Copenhagen, those bones sat in museum drawers for more than a century. Then, the age of ancient DNA arrived. In 2015, two independent research teams, one led by Eske Villerslev at the University of Copenhagen, another led by David Reich at Harvard, were working on ancient DNA from early Americans. They were separately analyzing skeletal material from different sites, using different samples, running different analyses, and they independently detected the same signal in the ancient South American material. It was a signal that pointed toward Australo-Melanesian populations, not Siberian, not East Asian, not ancient North Eurasian, but a genetic affinity with the ancestors of modern Aboriginal Australians, Papuans, and Andaman Islanders, populations that live on the other side of the planet from the Americas, separated by the Pacific Ocean, with no documented historical contact with the indigenous peoples of the New World. The signal appeared in a single individual from Lagoa Santa who died approximately 10,400 years ago. One person, one genome, one result that when it came back from the sequencing machines made the researchers stop. Jose Victor Moreno Meyer, who worked on the Copenhagen team's analysis, described the discovery in terms that are, for a scientist, unusually direct. How did it get there? We have no idea. David Meltzer of Southern Methodist University, one of the most respected Paleo-Indian archaeologists in North America, used a phrase that has since been quoted repeatedly in discussions of the finding. He described the Australasian signal as appearing to leap over all of North America in a single bound. It was present in one ancient South American. It was absent or undetectable in every North American ancient sample in the dataset. This is the detail that makes the finding genuinely strange. If a population carrying Australasian-related ancestry had entered the Americas through Beringia, the only confirmed entry route, that signal should appear in North American samples as well, at least at some sites. Populations moving southward through the Americas would have passed through North America first. They would have left genetic traces in the north before they left them in the south. But in the data available at the time, no such traces appeared in North America. The signal seemed to appear in South America without leaving footprints anywhere above it. The finding was not produced by a single lab using a single method. Two independent teams, working on different samples with different analytical approaches, detected the same pattern. That convergence is significant. When two independent groups using different methods find the same unexpected signal, the probability that it is a technical artifact drops considerably. What exactly the signal represents is a question that remains genuinely open. One interpretation is that it reflects a separate migration into the Americas, a second wave of people, or possibly a founding population that was more genetically diverse than previously understood, that carried Australasian-related ancestry into South America by a route that did not leave a detectable trace in North American populations. This could mean a Pacific coastal route that bypassed much of North America, or it could mean that North American populations carrying the signal were later replaced by other groups, erasing the genetic evidence in the north while South American populations retained it. Another interpretation is that the signal does not require a separate migration at all. It may reflect a founding Beringian population that was itself more genetically diverse, carrying Australasian-related ancestry as part of its initial genetic composition, and that this diversity was subsequently lost in most North American lineages through genetic drift, founder effects, or population replacement, surviving only in certain South American lines. Under this model, the Americas were not reached twice. The founding population simply carried more variation than the simplified models assumed. A third possibility, raised by some researchers, is that the signal reflects a very early coastal migration along the Pacific Rim, people moving southward along the western edge of the Americas, possibly by boat, much earlier than the mainstream models of initial entry, and carrying ancestry related to the populations of Southeast Asia and the Pacific that would eventually give rise to modern Australo-Melanesians. None of these interpretations has been definitively confirmed or ruled out. What has been established is that the signal appears in a small number of modern Amazonian groups, notably the Suru and the Caridiana, populations of the Brazilian interior, at detectable levels. It is not present in all ancient South Americans, and it is not widely distributed across modern Native American populations. Its geographic concentration in certain lowland South American groups, combined with its apparent absence from North American populations, is the core of the puzzle. The small group of modern Amazonian people who carry this signal are, in some sense, the living descendants of whatever ancient event the Lagoa Santa genome is recording. Whether that event was a separate migration, a diverse founding population, or something else entirely, they carry its biological trace into the present. and this matters for the broader question of what consumer DNA tests are measuring. The Australasian signal detected in ancient South Americans does not appear as a recognizable category on any commercial ancestry test. It would not show up as Australasian in a 23andMe result. But it is a reminder that the founding genetic diversity of the Americas was greater than the simplified reference panels assume. The people who eventually became the ancestors of all Native American populations carried a genetic legacy that was, in ways still being worked out, connected to populations across a much wider geography than Siberia alone. The detection of this signal also matters because it affects how geneticists understand the reference populations used to build the Indigenous American category in consumer tests. If the founding Native American population was itself genetically diverse in ways that include Australasian-related ancestry that is absent from the Latin American and Caribbean donors who form the core of the consumer test reference panels, then the reference panels are not capturing the full genetic range of Native American ancestry. They are capturing a subset of it. This creates a specific and largely invisible problem. A person with genuine ancestry from a Suru or Cariciana ancestor, one of the Amazonian groups that carry the Australasian-related signal, might receive a DNA test result that does not recognize their ancestry as indigenous at all, because the component of their DNA that reflects their native heritage does not match the algorithm's expectations for what Native American DNA looks like. The test cannot see what it was not trained to recognize. The Lagoa Santa genome is not a curiosity. It is evidence that the peopling of the Americas was more complex, more genetically layered, more migration diverse, more surprising than the model that emerged from the first wave of ancient DNA research. The Beringian hypothesis remains the dominant explanation for the origin of most Native American populations, and it is almost certainly correct in its broad outline. But the Lagoa Santa signal is a persistent anomaly that does not fit cleanly into that outline, and the researchers who produced it have said so clearly. For the question that started this investigation, why does a DNA test show Indigenous American ancestry for people with no family knowledge of Native heritage? The Lagoa Santa findings add one more layer. The category being measured by the algorithm is built on a partial understanding of the populations it is meant to represent. The reference panels are limited. The ancient genetic diversity of the Americas is still being mapped. And the map is not yet complete. The Australasian signal in a 10,400-year-old Brazilian cave is genuinely unresolved. The researchers who found it say so. The researchers who have tried to explain it have not reached consensus. It remains one of the most interesting open questions in the genetics of the first Americans. But while scientists work on the ancient past, a very different kind of confusion is playing out in the present, one that has less to do with Ice Age migrations and more to do with American mythology, family storytelling, and the strange persistence of a claim that appears in millions of families across the southern United States, the story of the Cherokee grandmother. Every family has its version. The details shift, the grandmother becomes a great-grandmother, the Cherokee becomes Blackfoot or Choctaw or Creek, the princess title arrives or is implied, but the basic structure of the story is remarkably consistent. Somewhere back in the family tree, there was a Native American woman. She was beautiful. She was proud. She married a white man, and her descendants became the family you belong to now. The proof is in the cheekbones. The proof is in the dark hair. The proof is in the way the family has always had a special connection to the land, or an affinity for the outdoors, or some other quality that the storyteller feels sets them apart. The story is so common that anthropologists have given it a name. The Cherokee princess myth has been documented, analyzed, and written about in the academic literature for decades. The anthropologist Joel W. Martin observed that an astonishing number of Southerners assert they have a grandmother or great-grandmother who is some kind of Cherokee, often a princess. The claim has been documented across families in Georgia, Tennessee, Alabama, the Carolinas, Virginia, and well beyond the historical Cherokee territory. It appears in families with no genealogical connection to the Southeast at all. It has spread geographically far beyond any plausible historical basis. Harvard University's Native American program has addressed the phenomenon directly in formal academic settings. Scholars have traced the myth's origins, its cultural functions, and its persistence across generations of American family storytelling. The conclusion is not simply that people are wrong about their ancestors, though many are. The conclusion is that the myth is performing a specific cultural work, and understanding that work requires asking why it exists at all. The Cherokee Nation's own position is unambiguous. There were no Cherokee princesses. The Cherokee Nation did not have a royal family, did not have a hereditary nobility, and did not use the term princess to describe any category of women in its traditional social structure. The title is a European imposition, a romanticization applied retrospectively by people who wanted to elevate the story of a native ancestor by assigning her a status she would not have claimed and probably would not have recognized. More practically, citizenship in the Cherokee Nation is determined not by DNA and not by family story, but by documented genealogical descent from a specific historical record, the Dawes Roll, a census of the Cherokee Nation compiled between 1898 and 1914. The Dawes Roll lists approximately 41,000 individuals who were enrolled as citizens of the 5 civilized tribes. Descent from a person listed on the Dawes Roll, traceable through documented genealogy, is the foundation of Cherokee citizenship. No genetic test, no family story, and no percentage on a pie chart substitutes for it. The Cherokee Nation currently enrolls approximately 370,000 citizens. Those citizens are connected to the tribe through documented, verifiable genealogy, through names and records, through paper trails that connect living people to people listed on the Dawes Roll more than a century ago. The Cherokee Nation is in its own governance, a community maintained through law, documentation, and shared history. Against this background, the 2010 United States Census reported that 819,105 people self-identified as Cherokee, more than twice the enrolled citizenship of all 3 federally recognized Cherokee nations combined. The gap between people who claim Cherokee identity and people who are documented members of a Cherokee nation is not a small one. It is enormous, and no amount of DNA testing can close it, because the question of tribal citizenship is not a genetic question. The myth persists for reasons that are genuinely worth examining, because they illuminate something about American identity that goes beyond genealogy. One reason is historical, real intermarriage did happen. Scots Irish traders who operated in the Cherokee territory of the 18th century Southeast did marry Cherokee women. The children of those unions existed. Some of them appear in historical records. Some of their descendants are enrolled Cherokee citizens today. The existence of the myth is not purely fiction. It grows from a seed of historical reality and then expands far beyond what the historical record can support. A second reason is social and psychological, claiming native ancestry, and specifically Cherokee ancestry, which carries particular cultural cachet in the American imagination, offers something to people whose family history is otherwise ordinary. It provides a connection to place, to land, to an older version of America that predates the histories of European immigration. For families whose ancestors were settlers or colonizers or simply farmers, the Cherokee grandmother offers a narrative of belonging that goes deeper than a boat crossing the Atlantic. It says, "We were here before we were here. We belong to this place." A third reason is more uncomfortable. Genealogical researchers have documented cases in which the Cherokee princess in a family story was, in fact, a light-skinned African-American woman, one whose racial identity was recoded in family memory after the Civil War, during a period when claiming native ancestry was socially preferable to claiming African ancestry in the white-dominated South. The family story preserved the memory of a woman who looked different, who did not fit the standard description, and reinterpreted her through a more culturally acceptable framework. The Cherokee grandmother replaced the black grandmother. The Indian features replaced the African ones. This is not a universal explanation, but it is a documented one, in enough cases to be taken seriously as a contributing factor to the myth's prevalence. For the people who take a DNA test hoping to confirm the family story, the results are often confusing. Some get a signal that matches the story, a small percentage of Indigenous American ancestry that seems to validate what they were told. Some get nothing at all. And some, as discussed earlier, are in the ambiguous middle, a signal that might reflect a real ancestor, might reflect an algorithm artifact, might reflect ancient Eurasian shared ancestry, and might have nothing to do with a Cherokee grandmother either way. But the myth's cultural dominance has a specific effect on the DNA testing industry. It has created a consumer expectation that Native American ancestry can be detected, confirmed, and quantified by a commercial algorithm. Companies have, in some cases, encouraged this expectation. 23andMe's own blog has published pieces discussing what it means to have Native American DNA, implicitly suggesting that the test can answer this question in a meaningful way. Kim Tallbear, an anthropologist at the University of Alberta and the author of Native American DNA, Tribal Belonging and the False Promise of Genetic Science, has written extensively about the consequences of this expectation. Tallbear observes that people who receive a small percentage of indigenous ancestry on a consumer test, 1%, 2%, 3%, mostly continue to identify as white in their daily lives. The result becomes an accessory to identity rather than a transformation of it. They do not join tribal communities. They do not seek enrollment. They do not take on the legal, political, or cultural responsibilities that actual tribal citizenship entails. They carry the percentage as a story, and the story does the same work the Cherokee princess myth has always done. The myth and the test result have become, in the 21st century, mutually reinforcing. The myth creates the expectation of a result. The result validates the myth, and neither one tells the people involved anything meaningful about the living nations whose identity they are, in some sense, borrowing. The Cherokee Nation's Secretary of State, Chuck Hoskin Jr., has addressed this directly and without ambiguity. A DNA test, he has stated, is useless for determining tribal citizenship. Current DNA tests do not even distinguish whether a person's ancestors were indigenous to North or South America. The test cannot tell you which tribe. It cannot tell you which nation. It cannot tell you whether the ancestor it is pointing toward was an enrolled citizen, a mixed heritage individual living on the margins of a community, or a genetic echo of a population movement that happened 10,000 years before the Cherokee Nation existed as a political entity. The myth endures because it fulfills a need, but understanding what the DNA test is actually measuring requires separating the need from the evidence, and the evidence, as it turns out, points in directions the myth was never designed to address. The saliva tube sits in a laboratory somewhere in California or Texas, one of millions processed each year by machines that read genetic code the way a scanner reads a barcode. The technician watching the output is not thinking about family myths or Cherokee grandmothers. The technician is watching data populate a screen, rows of SNPs, each one a location in the genome where variation between individuals is known to occur, each one being compared against a reference panel that the company has spent years assembling. The science behind consumer ancestry testing is real. The chemistry is real. The sequencing is real. And the statistical methods are legitimate tools of population genetics. But what the tests measure and what they claim to measure are not always the same thing. And the gap between those two things is large enough to explain a significant portion of the confusion that surrounds the indigenous American results. Consumer DNA tests, 23andMe, Ancestry DNA, Family Tree DNA, and others analyze what are called single nucleotide polymorphisms, or SNPs. These are specific locations in the genome where the DNA base at that position varies between people. The human genome contains roughly 3 billion base pairs, but the variation between any two unrelated people is concentrated in a subset of locations. Consumer tests analyze between 400,000 and 700,000 of these variable positions. That sounds like a large number, and it is. But it is also, as a fraction of the total genome, quite small, somewhere between 0.013 and 0.023%. The algorithm then compares the customer's SNP profile against a reference panel. The reference panel is a database of people whose ancestry is considered known, grouped by geographic region, self-reported ethnicity, or documented genealogy. And the algorithm asks, to which reference population does this customer's DNA most closely resemble, and in what proportions? The proportions become the percentages on the pie chart. The reference panel is the foundation on which the entire result rests, and the reference panel has a specific, well-documented problem when it comes to the indigenous American category. The people who built the major consumer genetic databases needed consenting participants who self-identified as having Indigenous American ancestry and who were willing to contribute their DNA to build the reference population. Obtaining those participants from enrolled members of North American tribes proved enormously difficult. For reasons that are historically grounded and entirely understandable, the Havasupai case is the most frequently cited example. Members of the Havasupai tribe of Arizona provided blood samples to researchers at Arizona State University beginning in the 1990s, under the understanding that the samples would be used to study diabetes, which was disproportionately affecting their community. The samples were subsequently used, without the tribe's specific consent, for research into schizophrenia, inbreeding, and population migration studies. The Havasupai sued. Arizona State University settled in 2010, returning the blood samples and paying $700,000 in damages. The case became a landmark in the ethics of genetic research with indigenous communities. It also became a reason. Many tribes, aware of the Havasupai case and of a longer history of extractive research practices, declined to contribute to commercial or academic genetic databases. The refusal was not irrational. It was a rational response to a documented pattern of exploitation, research that took biological material from indigenous communities, used it for purposes those communities had not agreed to, and sometimes produced findings that were used to undermine tribal claims about their history and origins. The result is that the consumer test reference panels for Native American populations are built largely without the participation of the people those panels are supposed to represent. 23andMe's North American Indigenous Reference Population is constructed primarily from people in Latin America and the Caribbean, individuals who self-identify as having indigenous ancestry and who are willing to participate. The specific genetic profiles of the Ojibwe, the Lakota, the Haudenosaunee, the Apache, the Navajo, populations that have been in North America for thousands of years and that have their own distinct genetic signatures, are dramatically underrepresented. This creates two specific problems that run in opposite directions. The first problem is false negatives. A person with genuine, documented, genealogically confirmed ancestry from a North American tribe may take a consumer test and receive 0 Indigenous American ancestry in the result. Their native heritage is real, their family history is documented, their ancestor is listed on a tribal roll. But their DNA does not resemble the Latin American and Caribbean donors who constitute the reference panel closely enough for the algorithm to detect it. The test misses the signal because the signal it is looking for does not accurately represent the population in question. The second problem is false positives, or more precisely, misclassified signals. A person with no recent Native American ancestry at all may carry genetic variants that resemble the Latin American indigenous reference population, either because of the ancient North Eurasian shared ancestry discussed earlier, or because of other ancient population overlaps that the algorithm cannot cleanly separate. The algorithm flags these variants as Indigenous American. The person receives a percentage. The percentage is not a measurement error in the technical sense. The variants are genuinely there, but the interpretation of what they represent is potentially wrong. Different companies give different results from the same raw data because they are using different reference panels and different statistical methods. A person who receives 2% Indigenous American from 23andMe might receive 0% from Ancestry DNA, or 1% from Family Tree DNA, because the three companies have built different models of what Indigenous American DNA looks like. None of the models is complete. All of them are approximations. Neneba A. Garrison, writing in the American Academy of Arts and Sciences journal Daedalus in 2018, summarized the technical situation clearly. Limitations of consistency across platforms and generalizability of knowledge remain significant challenges for genetic ancestry testing with indigenous communities. The tests are useful research tools. They are problematic identity documents. Ancestry DNA's own documentation acknowledges that it reconstructs indigenous ancestry by identifying small segments of DNA that resemble its reference population and then stitching them together into a percentage. The language is accurate and, if you read it carefully, quite candid about the approximation involved. The test is not reading a direct record of ancestry. It is making a probabilistic inference about what the ancestry might have been, based on a comparison to a limited and imperfect reference set. The companies are not unaware of these problems. They have been expanding their reference panels, adding more diverse populations, refining their algorithms, and adjusting their confidence intervals. 23andMe explicitly labels its ancestry estimates as estimates, not facts. The language in the fine print is careful. The language in the marketing is considerably less so. The practical consequence is that the consumer facing the pie chart on their laptop screen is receiving a result that is simultaneously based on real science and substantially limited in its interpretive power. The DNA is real. The analysis is real. The comparison is real. But the meaning attached to the result, that small percentage labeled Indigenous American, is the output of an approximation, built on a partial reference panel, shaped by decades of fraught history between genetic researchers and Native communities, and filtered through an algorithm that cannot distinguish between a Cherokee great-great-grandmother, an ancient Eurasian population overlap, and a Latin American indigenous reference donor whose genetic profile happens to resemble something in your genome. The test cannot tell you which of those things produced your result. It can only tell you that the signal is there. And for some people, the ones whose signal persists across multiple platforms, whose percentage is consistent across different companies using different reference panels, and whose genealogical records cannot account for it, the question of where the signal comes from leads somewhere the test cannot follow. It leads back into the historical record itself. Not the sanitized version, not the family legend, but the documented and underdocumented history of what happened in the Americas over the 4 centuries between first contact and the modern era. The history of people who mixed, erased, survived, and left a biological record that the paper record tried its best to remove. The frontier of colonial North America was not a line. It was a zone, broad, porous, constantly shifting, inhabited by people who did not fit the neat categories that official records preferred. In the interior of the continent, far from the coastal settlements that dominate the popular image of colonial history, a different kind of world was operating. It was a world built on trade, on kinship, on negotiated relationships between European newcomers and indigenous nations that had been managing this territory for thousands of years. The people who moved through that world left records, but the records are incomplete by design. Consider what a census form in the early 19th century was designed to capture: heads of household, numbers of family members, racial category, occupation, and property. It was not designed to capture the complexity of mixed heritage families navigating a society that assigned legal status and land rights based on race. It was designed to sort people into categories. And when the categories were inadequate for the reality, when the person being counted had a French father and a Natchez mother, or a Scots-Irish grandfather and a Cherokee grandmother, or a history that crossed the lines the census was built to enforce, the form chose a category and moved on. The form almost always chose white, if the person could plausibly be called white. The incentives for choosing white intensified dramatically over the course of the 19th century. The Indian Removal Act of 1830 authorized the forced relocation of Native American nations from their eastern homelands to territory west of the Mississippi. The implementation of the act, the series of forced marches that included the Cherokee removal of 1838, but also the removal of the Choctaw, Creek, Chickasaw, and Seminole nations, created an immediate and urgent reason for families of mixed Native and European ancestry to declare themselves unambiguously European. Removal applied to Native Americans. It did not apply to white people. The distinction was a matter of survival. Families made choices. Some enrolled with the tribe and went west. Some chose to remain in the east by documenting themselves as white, surrendering their tribal identity and, with it, the memory of that identity across subsequent generations. The children of those families grew up knowing themselves as white. Their children's children had no reason to remember otherwise. The story was gone before it could become a story. The 1887 Dawes Act added a different pressure. The act required Native Americans to enroll on official tribal rolls, to document their identity through a federal bureaucratic process in order to receive allotments of land. Families who did not enroll, for whatever reason, because they had already assimilated into white or black communities, because they distrusted the federal government's intentions. Because they lacked the documentation the process required, were excluded from the rolls. Their descendants had no enrollment record. Without enrollment, their native ancestry became, officially, invisible. The Bureau of Indian Affairs blood quantum system, which required people to demonstrate a minimum fraction of native ancestry to qualify for certain federal programs and rights. Added yet another layer. A person with one Native American grandparent and 3 European grandparents might not meet the blood quantum threshold. Their children, with less native ancestry still, certainly would not. Over several generations, the legal identity dissolved even when the biological one persisted. The result of these overlapping pressures: removal, assimilation. Enrollment requirements, blood quantum, was a systematic narrowing of who was officially recognized as Native American, and a corresponding expansion of the population that had native ancestry but no official recognition of it. The genetic signal survived the legal erasure. The DNA does not require a Dawes roll entry. This history is particularly relevant for African American families. The relationship between Black and Native populations in North America is one of the most complex and least discussed aspects of American demographic history. Enslaved people and Native Americans shared territory in ways that produced sustained contact, kinship, and biological mixing across centuries. In the Southeast, where both populations were concentrated before the removal, the proximity was especially close. Maroon communities, groups of escaped enslaved people who established free settlements, sometimes formed in or adjacent to Native American territory, and sometimes with Native American participation or protection. The Seminole Nation of Florida is perhaps the most well-documented example. The Seminoles incorporated significant numbers of Black people, both escaped slaves and free individuals, into their communities over generations. The descendants of those relationships are sometimes called Black Seminoles, and their history is a compressed version of a process that happened less visibly in many other contexts. After the Civil War, the 5 civilized tribes, which had held enslaved people before the war, were required by their treaties with the United States to extend tribal citizenship to their freed people. The freedmen of the 5 tribes have been a source of ongoing legal and political dispute, particularly as tribal nations have, in some cases, sought to restrict their membership. The genetic ancestry of Black freedmen descendants often includes significant Native American components, ancestry acquired not through a mythologized grandmother, but through generations of living inside tribal communities. The 23andMe data, showing that over 22% of African Americans carry at least 1% Indigenous American ancestry, is not a mystery when viewed through this history. It is the expected biological outcome of centuries of documented contact, cohabitation, and kinship. The surprise is not that the signal is there. The surprise is that it was not better documented before the genomics data made it visible. For European American families, the hidden history runs through a different set of channels. The fur trade era of the 17th and 18th centuries produced enormous numbers of mixed heritage families across the Great Lakes, the Upper Mississippi, the Ohio Valley, and the Southeast. The Imptis people of the Northern Plains and Canada, descendants of French, Scottish, and English traders, and the Cree, Ojibwe, and other nations, represent the most visible legacy of this mixing. But they are not the only one. South of the Canadian border, the same processes produced mixed heritage families who assimilated into the broader white population rather than forming a recognized distinct community. In Louisiana, the French colonial period produced a 3-way mixing of French, African, and Native populations that was encoded in the Creole culture of the lower Mississippi. The Natchez, the Homa, the Tunica Biloxi, the Caddo, and other nations of the region were deeply embedded in the colonial economy for more than a century before American sovereignty arrived and began sorting people into the black-white binary that Louisiana's own culture had never been organized around. The sorting erased the middle. The signals in the DNA data are, in many cases, what survived that erasure. They are the biological persistence of a history that was, for practical and sometimes life-saving reasons, buried in the official record. But there is a crucial distinction that the DNA cannot make. It cannot tell you whether your signal comes from a fur trader who married an Ojibwe woman in the 1720s. or from an ancient population overlap between Beringian founders and Proto-European ancestry that occurred 20,000 years before any fur trader existed. It cannot tell you whether your ancestor was an enrolled tribal member or a person of mixed heritage who was never counted by anyone. It cannot tell you the name, the nation, the community, the language, or the lived experience of the person whose genetic material found its way to you across generations of mixing, erasure, and chance. The DNA is the residue of history, not history itself. And for the living nations whose identity is implicated in these results, the distinction between residue and history is not a semantic one. It is the entire question. In 1978, the United States Congress passed the Indian Child Welfare Act. The law was a response to a documented crisis. For decades, the estimates suggest from the 1940s through the 1970s, between 25 and 35% of all Native American children were being removed from their families and placed with non-Native adoptive families or in non-Native foster homes. Some estimates put the figure higher. The removals were carried out by state welfare agencies, operating under assumptions about Native family structures and child welfare that reflected the assimilationist policies of the era, rather than any genuine concern for the children involved. The children who were removed grew up in non-Native families, in non-Native communities, without knowledge of their tribal nations, their languages, their relatives, or their histories. Many of them were told nothing, or were told something false. Some were told their birth family was dead. Some were told they were white. Some were given new names. They grew into adults with a gap where their origin story should have been. Some of those adults are alive today. Some of them have taken NA tests. For those individuals, the Indigenous American signal on a consumer ancestry test is not a family myth or an algorithm artifact or an ancient Eurasian genetic echo. It is evidence of something that was taken from them. And the test, for all its limitations, is sometimes the first piece of evidence they have ever held. This is the most human dimension of the question that began with a kitchen table and a pie chart. Behind the statistics, the 8% in Louisiana, the 22% of African Americans, the haplogroup X2A frequency in Algonquian populations, there are people for whom the result is not an interesting puzzle, but an urgent personal mystery. People who were separated from their communities by policy, by poverty, by force, and who are now trying to reconstruct a history that was deliberately destroyed. The Indian Child Welfare Act was designed to stop the practice by requiring that child welfare proceedings involving Native American children give preference to placement with extended family, tribal members, or other Native families. The law has been controversial and has faced legal challenges, including a case that reached the Supreme Court in 2023. But its passage in 1978 acknowledged, officially, that something systematic had been done, that a generation of children had been separated from their origins as a matter of policy, and that the consequences were real and ongoing. For those individuals and their descendants, the consumer DNA test is doing something that the test was not designed to do. It is functioning as a witness. It is producing a number that points toward a history that no one told them. It cannot give them a name, a nation, or a community, but it can give them a direction. And here is where the technology's limitations become most important to understand and most important to treat with care. The test can produce a signal. It cannot produce belonging. It can say that some fraction of the DNA in your cells resembles the reference population the algorithm has defined as Indigenous American. It cannot say which nation, because the algorithm cannot distinguish between Ojibwe and Navajo and Lakota and Seminole. It cannot say which ancestor, because genetic ancestry does not come with names attached. It cannot say whether the person whose DNA you carry was an enrolled tribal member, a mixed heritage individual living at the edge of a community, or a person whose native heritage had already been erased before it reached them. Kim Tallbear's analysis of this situation is the most precise available in the academic literature. Tallbear distinguishes between genetic ancestry, the biological residue of population history, and tribal belonging, which is a legal, political, cultural, and communal relationship that is determined by tribal nations themselves, not by DNA companies. These are not the same thing. They have never been the same thing. But the consumer ancestry industry has, in many cases, blurred the distinction in ways that cause real harm. The harm operates in two directions. For tribal nations, the proliferation of DNA-based identity claims creates political and legal problems. If a person can claim Cherokee identity on the basis of a consumer test result, and if that claim gives them access to programs, preferences, or recognition intended for enrolled tribal members, then the test result is doing political work that tribal nations did not authorize and cannot control. Tribal sovereignty, the right of a nation to determine its own membership, is undermined by the suggestion that a commercial algorithm can substitute for a nation's own enrollment criteria. For the individuals receiving the results, the harm is subtler, but equally real. The test can create the feeling of belonging where the substance of belonging, community, relationship, language, shared history, mutual obligation, does not exist and cannot be generated by a percentage. A person who receives 3% Indigenous American on a DNA test and subsequently claims Native identity has not gained what they believe they have gained. They have gained a number. The number points towards something, a real history, a real ancestor, a real connection to the populations that built this continent over thousands of years. But the number is not that thing. It is a shadow of it. The Dawes Roll is a document. The Indian Child Welfare Act is a law. The enrollment criteria of the Cherokee Nation, the Navajo Nation, the Oglala Sioux tribe, and every other federally recognized tribe in the United States are expressions of sovereignty, the right of those nations to define who belongs to them. None of that is captured in a pie chart. And yet the pie chart points somewhere real. The DNA that produces the result came from somewhere. An ancestor lived somewhere, belonged to something, spoke a language, maintained relationships that the DNA algorithm cannot see. The test result is not an identity. But it is not nothing either. It is evidence, partial, probabilistic, often ambiguous, of a history that has been incompletely documented by the people who were most invested in not documenting it. The Indian Removal Act created erasure. The Dawes Act created bureaucratic barriers. The forced adoption era created literal orphans of culture. The genomics data is in some cases the only surviving trace of what was lost. It cannot restore what was taken, but it can confirm that something was there. For the living tribal nations, none of this changes the fundamental question of membership. The Cherokee Nation's position is clear. A DNA test is useless for determining tribal citizenship. This is not a policy disagreement. It is a statement about what tribal citizenship is. It is a political relationship, a cultural commitment, a genealogical documentation, not a biological measurement. The test can point toward an ancestor. Only a community can offer belonging. For the individuals who receive unexpected results and begin asking questions, the test is often the beginning of a longer process. A search through genealogical records, a conversation with family members, a request to a tribal enrollment office, an inquiry into a history that was obscured across generations. Sometimes that process leads somewhere. Sometimes it leads to a gap that cannot be closed, a record that was destroyed or never made, a branch of the family tree that ends without a name. The signal on the test is real. The meaning of the signal is not something the test can provide. Return to the kitchen table. The laptop is open. The pie chart is on the screen. The small sliver, labeled Indigenous American, is glowing in the quiet of whatever hour this is. Late evening, maybe, or early morning before the rest of the house wakes up. The number is the same as it was at the beginning. 2%, maybe 3%. The saliva tube is long gone, processed by a machine in a laboratory that will never send it back. But the person sitting at the table is not the same as the person who mailed the tube 6 weeks ago. They know now that the number was produced by an algorithm comparing their DNA against a reference panel built mostly from people in Latin America and the Caribbean, assembled with the participation of very few enrolled members of North American tribes, because the history of genetic research with Native communities made participation, for many tribes, a rational thing to refuse. They know that the algorithm they are looking at cannot distinguish between a Cherokee great-great-grandmother in 19th century Georgia, An ancient population overlapped between the Beringian founders and Proto-European ancestors of some of their own ancestors, and a Latin American indigenous donor whose genome happened to resemble something in their own genetic code. They know that two siblings with the same family tree can get completely different results from the same test. Because chromosomal inheritance is random, and by the 5th generation, a specific ancestor's DNA may simply not have been passed down to you at all, or may have been passed down in fragments that the algorithm catches where it misses in your sibling. They know that the signal is stronger in Louisiana than in Minnesota because of a specific history, French colonial trade networks. Natchez and Homa intermarriage, a three-way mixing of populations that the American racial binary later tried to sort into two boxes. They know that over 22% of African Americans carry a detectable indigenous signal because the history of Black and Native contact in the Southeast was deep, sustained, and produced real biological kinship that the official record was never designed to capture. They know about the ancient North Eurasian component, the Malta genome, the 24,000-year-old individual from Siberia whose DNA revealed that Native Americans and Europeans share a distant common ancestor, a lineage now called Ancient North Eurasian that left its trace in both populations through completely different routes. They know that this ancient overlap can appear in a European-descended person's genome and be classified as Indigenous American by an algorithm that cannot distinguish ancient shared Eurasian ancestry from recent Native American descent. They know about haplogroup X2A, the mitochondrial lineage present in up to a quarter of Algonquian populations found in Kennewick Man. Confirmed as pre-Columbian, whose Old World parent population has never been identified in any living population and probably went extinct during the Ice Age. They know about the Solutrean hypothesis that tried to use X2A as evidence for an Atlantic crossing, and they know that it collapsed when the ancient DNA from actual Solutrean individuals showed no relationship to Native Americans at all. They know the lesson of that collapse. Genetic patterns can be misread dramatically by serious researchers when the right ancient samples have not yet been tested. They know about the bones from Lagoa Santa, the Brazilian cave, the 10,400-year-old individual, the Australasian genetic signal that two independent research teams detected and that no one has fully explained. They know that Jose Victor Moreno Mayor said plainly that we have no idea how that signal got there. They know that the founding genetic diversity of the Americas was greater than the simplified models assumed, and that the reference panels used to produce the pie chart on their screen do not capture that diversity fully or accurately. They know about the Cherokee Princess myth, the anthropological documentation, the Harvard lecture, the Cherokee Nation's explicit statement that there were no princesses, and that citizenship is determined by documented genealogy, not by story or percentage. They know that the myth performs cultural work, that it provides belonging to people whose connection to the land feels insufficiently deep, and that it sometimes conceals a different kind of hidden ancestry that the storytellers found less socially acceptable to claim. They know about the Indian Removal Act of 1830 and the incentive it created for families to erase Native identity from the official record within a single generation. They know about the Dawes Roll and the families who were excluded from it for reasons that had nothing to do with their actual ancestry. They know about the Indian Child Welfare Act and the generation of children who were separated from their tribal communities by policy and who grew into adults with DNA that points toward a history nobody told them. They know about the Havasupai case, the blood samples taken for diabetes research and used without consent for studies on schizophrenia and migration, the lawsuit, the settlement. The reason that many tribes have declined to participate in the databases that would have made the reference panels more accurate. They know that the absence of North American tribal DNA from those databases is not an oversight. It is the result of a relationship between genetic researchers and indigenous communities that was damaged by exploitation and has not been fully repaired. They know that the Cherokee Nation's Secretary of State stated clearly that a DNA test cannot determine tribal citizenship, and that current tests cannot even distinguish between ancestry from North and South America. They know that Kim Tallbear drew the distinction between genetic ancestry and tribal belonging, that belonging is a legal, political, cultural, communal relationship that tribal nations determine for themselves, and that no algorithm produces. The number on the screen, 2%, 3%, 1%, is real in the sense that the DNA is there, the variants are there, the comparison was made, the output is not fabricated. But what the number means is a question that runs through ancient Ice Age migrations, colonial frontier marriages that were never recorded, deliberate erasures of identity across multiple generations of American history, the incomplete and politically fraught construction of commercial genetic databases, and the ongoing sovereignty of living nations who have their own answers to the question of who belongs to them. The past was more interconnected than the clean story allows. Human populations moved, mixed, separated, and mixed again across timescales that make the entire colonial era look recent. The people who eventually became the founding population of the Americas carried genetic traces of populations that no longer exist, that left no modern descendants except through the lineages that crossed into a new world and diversified across 2 continents over thousands of years. Some of those traces are still detectable. Some of them look, to a commercial algorithm, like a small percentage. The number is real. Its meaning is not something the test can give you. Outside the kitchen window, the same geography that produced the signals in the data is simply there. The land, the watershed, the elevation, the same terrain that the Natchez and the Cherokee and the Ojibwe and the Lakota and every other nation that ever existed on this continent organized their lives around for thousands of years before anyone thought to ask who was here. The past that produced the signal on the screen was lived in specific places by specific people whose names were not always written down, and whose relationships were not always recorded, and whose descendants did not always know what they were carrying. The laptop closes. The pie chart disappears. The small percentage is still there, encoded in the cells of the person who is now getting up from the table. The past still keeps something back. It kept the names. It kept the languages that were spoken in the places the signals point toward. It kept the specific face of the ancestor the algorithm detected and then declined to identify. The DNA survived. The context did not fully survive with it, and that gap between the signal and the story, between the percentage and the person, between the biological trace and the living community it points toward, is not something any test will close. It is the condition of trying to read the past through the present, when the past was more complicated than the present was built to remember.